Hello, everyone, and welcome. My name is Rosie Manser, and I'll be your moderator for today. We're going to be talking about Plasma-FIB technology and the wealth of possibilities that this provides for life science research. I'm really grateful to have had the chance to speak with a number of different researchers about their journey to Plasma-FIB technology. Many of them have shared their experiences and research highlights associated with the Hydra Plasma-FIB system, and it was great to hear about the really wide variety of experiments that the system is being used for. I also had the chance to speak about the technology with Sveta Tomova, who is the product marketing manager for the Hydra at Thermo Fisher. There will be a live Q&A session at the end, and I'll be joined by some of the researchers who are involved in this event today. So please do ask questions throughout using the Q&A box, and we'll address as many as we can at the end. So to start things off, I began by asking Sveta about FIB technology in general and how it works.
FIB-SEM technology, or focused ion beam scanning electron microscopy, is a key technology in life sciences. It's actually well established, and it's the technology to go when you'd like to visualize ultrastructure at a high resolution and in 3 dimensions. How it functions, it's in the most simple way. It is automated milling of a sample surface. And milling means removal of a thin layer of a sample. When this new layer is exposed, or new surface is exposed, then the electron beam is imaging at a high resolution in a consecutive manner. So you just have milling, imaging, which ultimately gives you data sets that can be reconstructed into a 3D volume. FIB-SEM can be used conventionally for classical fixation or chemical fixation in resin-embedded samples, but it's very powerful when you want to thin cryo-fixed hydrated samples to manufacture a thin lamella that could subsequently be visualized and imaged at the TEM, or you can collect a 3D volume from your hydrated samples at high resolution.
Fantastic. So a pretty flexible technology for doing lots of different things. So could you give us a flavour now of the different types of FIB-SEM instrument that are available?
The most commonly used and well-known are the gallium FIB-SEM systems. Recently, especially on the life science community, was getting acquainted with the plasma-focused ion beam technology. And maybe the most important difference compared to the gallium is the fact that the plasma beam is collimated even when you go to very high currents. Why this is advantage and why this is attractive is simply because you can now remove faster and larger volume. And again, the question why this should matter is because now you can access your region of interest either for visualization or for extracting it for preparation of a lamella or lift outs much faster. So you're way more efficient. And I think this is the key behind plasma technology, what attracts not only life scientists, but recently also life science users.
It's exciting to hear how plasma offers some different opportunities over gallium. As part of putting together this event today, I spoke to a number of different key users who are already using the Hydra Plasma-FIB. And one of those users was Sebastian Tacker at the Max Planck Institute in Dortmund. Now, he specialises particularly in lamella prep as part of his work. And I was asking him generally, from the Hydra perspective, how this supports the research needs and requirements at the Max Planck.
I'm responsible for the hardware research, so my research focuses on development of new tools, techniques to increase the throughput of the entire cryo pipeline. Here in the Institute, we work on several fields. So, for example, in Department 1, Andreas Musacchio, he's working on the kinetic core system. And Department 3, we're working on different kinds of muscle to understand muscle function in a healthy state, but also when the system is somewhat disturbed during sickness. And we are not only working with isolated cells or microfibrils, we are also working with tissues, organoids. And as you can imagine, such systems are quite thick, and you need to thin them down so that they are electron transferred. So FIB-SEM technology is the most important tool here or technique which we use to thin our samples for tomography, because we believe tomography is one core technique because we have the ultra-structural landscape, but we can also dig deeper and get the structure of the proteins in situ by subthermal averaging.
So for Sebastian, it really is all about lamella prep for cryo-electron tomography. And during our conversation, which was fantastic, I kind of asked him about his journey and the technology journey that he's been on to be able to generate the best quality lamella.
When we started, I think 8 years ago with the entire preparation work, we started, of course, with isolated cells or myofibrils, and it was pretty accessible also with the cryo-FIB technology these days to prepare lamella. They are quite thin, they can be plunge frozen, and with automation we were really able to get a high throughput of lamella per day, and also the data sizes increases from several tomograms to some hundreds of tomograms. And this is of course quite useful for us since we need quite a lot of data to do subthermal averaging. But of course, we shifted then our focus more into tissues and organoids because they are more clinically relevant. And by nature, as I said, they are much thicker than isolated myofibrils. So how do we deal actually with the increased thickness? So the material we need to remove for the lamella production. The Hydra gives us the opportunity with the high currents and the xenon ion source that we can build at high throughput. Because initially, when this technique came up, lift outs were limited to a few per day. And with the high currents, we are now able to lift out much more volume per day. And I would say the subthermal averaging analysis is a numbers game. You need quite a lot of numbers, particles, to average these, to increase the signal-to-noise ratio. And in this sense, throughput is one of the main key features we need to keep high. So the Hydra is really the instrument which helps us here quite a lot.
Sounds like huge progress over the course of 8 years. And from what Sebastian was saying, it really seems like an improved throughput in lamella prep really is what results in an improved scientific output.
Well, throughput is a key word, but I believe quality of the lamella ultimately is what matters most.
Tatiana, who works with Sebastian at Max Planck, was also talking about quality. A lot of her work is based around being able to optimize the quality of the lamella that come from the systems. And it was great to talk to her about her perspectives on technology and how that's helping to improve lamella prep techniques.
I joined the Max Planck Institute specifically to work on the PFIC, to look into the damage that happens when you use the very high currents that are available on a plasma-FIB that are not available on a gallium-FIB. And so I'm really focusing on how badly can we treat our sample in terms of current levels without introducing any damage into the sample. And now that we are wrapping up a big step of the project and have a very good idea what we can do in terms of current usage without affecting our sample, I'm now assisting my colleagues that work with all sorts of samples, mostly different types of tissue samples, that now want to investigate these samples in a timely manner with high throughput and have them setting up their lift-out experiments and just getting them up to speed on the Plasma-FIB to really take advantage of this very, very beautiful instrument.
I was talking to Tatiana about the technology specifically, Plasma-FIB technology, and her opinion on how that's really changing this lamella prep workflow, and also how her colleagues react when she kind of introduces them to this new technology.
It's a big word, but I think it's going to be a game changer. Since with gallium-FIB, there's only so much milling you can do in a given amount of time, and we all know that time is super precious, and you want to have high throughput for statistics. And with Plasma-FIB, it's a completely different time scale. Like things that would in the past with gallium-FIB take overnight milling, maybe milling almost for 24 hours. This can now be done with Plasma-FIB in a super short amount of time. And we are talking here like half an hour, with these extremely high currents that are available on the Plasma-FIB. Whenever I show new colleagues to the Hydra that maybe have some experience already on the Aquila system with gallium-FIB, on the one hand, they are just amazed that it basically feels the same, since the user interface of the software is almost identical. Like the main buttons are there where you expect them to be, and then it's just really specific differences related to whether you have either the one type or the other type of source. So switching over from one type of instrument to the other one is super smooth. On the other hand, they're just absolutely happy and amazed. Oh wow, you want to mill this tremendous trench? Isn't that way too big? And then you just increase the current, and they're like, oh my goodness, it's so fast, it's so fun. So switching from FIB to Plasma-FIB is super easy, super smooth.
I really love it when a technology makes such a difference to a workflow. It's fantastic to see the impact that it's having. Before I left the team at Dortmund, I actually asked Sebastian about his perspectives on how Plasma-FIB is really changing the face of biomedical research and the potential that it holds.
From my point of view, the impact of, let's say, Plasma-FIB technology on the medical field, I think can be quite huge. If you imagine for our current research, we use like isolated cells, isolated microfibrils, easy to know. But most of the relevant questions, they don't occur in isolated systems. So they occur in complex systems. For example, if you want to research the neuromuscular junction, there are already two different cell types which need to interact. And often diseases occur at this points where two different systems connect. And due to the complexity of such systems, we need to think about preparation methods to bring them down to be electron transparent. And there, of course, since these are thick samples, organoids, tissues, it's really important to have a method which provides a decent throughput, or a throughput where you still can create enough data to do subtomogram averaging. Therefore, for me, it's obvious that Plasma-FIB technology is a key instrument to examine medical or clinical effects in the world of biology.
One of the other things that Tatiana and Sebastian also talked about was the use of different ion species for Plasma-FIB for being able to do different things. Could you give us an overview of kind of what the options are?
Well, you know that with Hydra you have the choice of 4 ion species. This is the xenon, oxygen, nitrogen, and argon. And why this is important, why the flexibility of choosing an ion species is important, is because you can adjust your experimental setup and use the most appropriate beam for the specific sample. And what I mean with this, I mean, it's usually it takes an effort to optimize sample preparation to make it compatible with the electron microscope, regardless of which microscope, but especially for FIB-SEM imaging. However, this compromises the initial biological question. And now, by having this flexibility, you can actually choose the sample that will suit best the biological question and not try to optimize the sample to suit your EM. And therefore, the flexibility of those of having different ion species is extremely beneficial. For example, when you look into low-acryl resins like LR-White and low-acryls, HM20, those are usually used especially when you want to benefit from preservation of fluorescent signal in the sample. But those resins are kind of prone on artifacts when using the gallium FIB-SEM tool. Therefore, they're generally avoided. However, if you look at this example with the brain tissue, we used oxygen and LR-White, and the results are just fabulous.
It's a really key point about what you said with the system being able to adapt to the specimen rather than the specimen adapt to the system. It's something also that I heard from the team at the Rosalind Franklin Institute, who I was lucky enough to have a great conversation with a few weeks ago. Tom Glynn is the PFIB manager there, and I started off by asking him about how FIB technology in general supports the work at the Rosalind Franklin Institute.
At RFI, it's a very exciting place to work. We do a lot of, obviously, life science, but we also do technical development. And one of the main things that we've been working on here is to be able to take tissue samples and go from tissue length scales down to the kind of nanometer resolution that we can get to using tomography techniques.
This capability that Tom refers to of being able to work across length scales in different life science specimens, is this something that you're hearing a lot from different user groups?
Visualizing biology in its complexity is a multi-scale approach. From having to look into molecular complexes in their natural environment, all the way to utilizing volume microscopy to have those fabulous 3D sets of the whole cells or tissues. This is all biology, and I believe Hydra Bio is like pushing the limits and breaching those scales and resolutions.
I completely agree with you. And from the perspective of the RFI, I actually asked Tom specifically about PFIB technology and how he sees it kind of being able to move the field forward, as you say.
Gallium is great and has been great for a long time, but one of the drawbacks of gallium is that it takes a long, long time to mill large volumes. When I was first trained to use the FIB, I was told, if you summed up the volume of everything that's been milled ever, it'd be much less than a millimeter cubed. And that kind of stuck with me because it's kind of a little bit mind-blowing. Sum up all of the FIBs all over the world throughout history in such a tiny, tiny volume. And I don't think that's true anymore. And maybe plasma is maybe partially the development with the plasma FIBs is really part of that, I think. And that's very exciting because that was really one of the big drawbacks with gallium.
I was also lucky to speak to Thermo, who is a group leader at the Rosalind Franklin Institute and is an expert across all different types of cryo-microscopy. And I asked her initially about her perspectives on how the Hydra Bio generally supports the work at the RFI.
Having the Hydra is really an ideal system for an institute like the Franklin. The Franklin is based on technology development with a big impact for human health. And so for that, we need equipment that have enough flexibility and a team on the back of it that have enough trust in us. So we can modify our equipment, we can push them to the limit. And on this journey, it's been very interesting because the Hydra is our tools to go when we want to do something new. It's got a big chamber, very easy to control stage, a bunch of fantastic different detectors, the possibility to welcome externals interface. It's a machine that is used for material science, so the perspective are endless. It's very well characterized in material science, so our job is somewhat easy in a sense that it's not completely new, in the sense that the machine is well characterized, but it's new for biological samples.
FIB technology is well established already in material science, and we can benefit and use this knowledge to tailor and optimize the workflows for life science. Really good point from
Really
Maud.
great. Collectively learning to be able to push the technology spaces forward. So I also spoke to Maud about the different specimen types that they have at the RFI. They work with everything from cells through to tissues with all sorts of different requirements and applications. And she spoke about some of the work that they're doing with these very varied sample types.
Or the project that I talk about will require one or another to be shaped. So you need to be shaped into either to give access to a sublayer of your system that you're working on, your biopsy or your cells or your organoids, or you need it to be shaped so it can fit into the microscope, which is the limitation that we have at the moment. And so being able to use a plasma ion beam rather than the more traditional gallium beam allow us to increase our speed. We also have a choice. We have a choice of 4 sources, so we can really adapt to your samples. So the approach that we have in here that have been very important, and I think this is why we like so much having the Hydra, is we're trying to find the best parameter for the samples. Rather than trying to fit the samples to the requirement of the microscope, we're trying to make sure that this is a microscope that adapts to the samples. And we have characterized, for example, the effect of the argon beam or xenon beam, as well as oxygen and nitrogen to a lesser extent on our biological samples. So we have demonstrated that in our hand, and we have better curtaining rate or less curtains when using argon on vitrified biological samples, that xenon is our faster beam, and that you can combine them to do the speed of xenon and the smoothness of argon. And having a smooth surface is very important, because if you have that, you can now unlock volumetric SEM imaging where you don't have curtaining artifacts. What we've been left with is charging artifact, and thanks to some work that we have done, we are now able to control it, which now we can reach the space with this Helios Hydra that gave us all of this flexibility to do volume SEM imaging without any artifact.
I think we still need to improve on this aspect, but on the other hand, it shows the potential and the flexibility of the platform, that people can really write their own scripts and look into options how to push the limits.
Absolutely, and absolute testament to the team at RFI for all the great work that they're doing on taking the technology and developing it even further. So we've talked a lot about the cryo aspect of the Hydra, the lamella preparation, the volume imaging, and the great potential there is there, but that's not the only thing that the Hydra Bio does, right? There's also the volumetric imaging in resin-embedded room temperature specimens. Can you tell us a bit more about that, Sveta?
Of course, we touched a little bit on the power of plasma to access large volumes, and this is obvious for the classical cross-sectioning approach, but Hydra Bio is also offering a novel application, which we'll call it spin mill. And the power of this is that now you can access the sample in its horizontal plane by milling at glancing angles. And this allows you to open a very large area of almost 1 millimeter in diameter, and then subsequently image the areas of interest. While we call this spin milling, is while the sample is milled, it's also being rotated at predefined positions, and therefore the name of the spin milling. I'm very often asked why spin milling. But coming back to the benefit of this approach, it's similar to what you might know from serial block phase imaging. So you really remove the material and then image the sample, and you create again a stack of images that ultimately will serve for the reconstruction of the 3D volume. What is beneficial of having access to this large area is that you immediately can identify the sparse regions, which are the regions of interest and where you actually would like to record at high resolution your volumes. And the other aspect is that you can combine spin mill with multi-imaging facets. That means that for each region of interest, you might use a different imaging settings. So you might have an overview at a lower resolution, and then you just can zoom in and have a sub-volume at a super high resolution. It's really very flexible, and I think this can push further the options for generating more statistically validated data, because now very easily can compare different subsets of features, or you might really compare treated against not treated samples.
I mean, it sounds fantastic in principle to be able to capture all these different regions at high resolution and have this huge field of view, but surely that leads to challenges in terms of acquisition time to start with. It must take a long time, and also the data size.
You're absolutely correct. One of the most limiting factors for FIB-SEM technology to be broadly accepted is exactly the time it takes to get to your data. And the limitation here is, of course, the scanning, the imaging part. So this is true for the gallium, and this is true for the Plasma-FIB. And this is where we recently adopted an AI approach to consider different scanning models. So we introduced the so-called adaptive scanning, which means that you can use a dual scanning mode, low, high resolution, for a given data set or for a given field of view. The benefit is that you don't always need the contextual information at the high resolution that is required for the area of interest, but you still need this contextual information. Sometimes you have cells or organisms which are surrounded by just the resin that they're embedded in. And this resin does not contain any information, but you still, in the conventional way of scanning, you still do scan with the selected high resolution. So by using the artificial intelligence, you create a map or a mask of your region of interest, and you kind of teach the system to recognize the region of interest against regions that are less important or do not contain information. So at the end of your acquisition, you are having a volume that has the contextual, low-resolution information and traces your features of interest or regions of interest at the desired high resolution, which saves significant amount of time. You can save up to 50%, and this is really a game changer. So you can have either more volume or you can have more volumes if you really need to build up these statistical validations.
How hard is it to train these networks and to actually configure the system to recognize the features that you want to image?
I can show you. So it's really easy. We already have implemented this option within the Auto Slice and View software. And the tool works the following way. You have a pen, and you can even decide what would be the thickness of the pen. This pen will be used for the annotation of the features of interest. And what we are looking here is cells. These are trypanosoma cells, and they're embedded in just empty resin. Once we have selected the pen and the thickness that we want to use, we can start to annotate the features of interest. First, we will contour them so that you feel comfortable, and there is no problem. As you have seen, you can always go to the eraser and you can always correct if something is not really optimal and you have any doubts. So once you complete the contouring process, then of course you can go safely and just really annotate the entire region of interest. Okay, so again, you're not limited. You're free to further correct and adjust, maybe delete an option. But once you're happy, you just submit the annotation that you have done. And this is what the tool will use to create the mask. So the mask is still, you can adjust it by using those plus and minus options to further tailor and adjust specifically the mask to your liking. And once this is done, you just continue the job and the AI will do it automatically. That means that you will acquire slice by slice at high resolution the area that you annotated and selected as your region of interest, while the rest is connected at low or is acquired at low resolution. Ultimately, you're having the volume with all of the information. High resolution is your area of interest. Low resolution is the contextual information. In this particular case, just empty resin.
That all looks really quite straightforward. As often with these big imaging instruments, it's not until you get the systems in the hands of researchers who are actually using them that you really troubleshoot and have a look at how the technology works. And to that end, we're now going to hear from Kirk Schimmick, who is at the Donald Danforth Plant Science Centre. He is a group leader there, and he also runs the Advanced Imaging Centre. And he worked closely with us in the development of this product, the adaptive scanning product, and he was one of the early adopters. So I started off by asking Kirk about his research interests and what happens at the Donald Danforth Centre.
A lot of our work requires high-resolution imaging of subcellular components in plants and the microbes. And we have transmission electron microscopy, actually a Telos 120, which is great for a lot of our high-resolution work. But the reality is our biology is a considerable three-dimensional element to it. So we recently acquired a Hydra focused ion beam, plasma focused ion beam scanning electron microscope to be able to extend our knowledge from more two-dimensional into larger three-dimensional volumes. One of the challenges that our lab faces is that we're interested in looking at the cell cytoplasm in plants. And it turns out, if you ever looked at a plant closely, about 90% of a plant cell is a large vacuole in the middle of the plant. So we're scanning large areas with not as much content of interest to us. Second thing is, when you're working with leaf tissue, there are air spaces. So all of these add up to time of scanning at high resolution when you're really only interested in this, 10% strip around the outside of the cells. But up until now, we've been forced to scan everything at high resolution if we wanted to see all that detail.
Really quite a familiar story to the trypanosomes that you showed in the software example. So we also asked Kirk, from the adaptive scanning perspective, what difference that makes to him when he uses the software.
With very easy drawing tools to go in and outline, the cell cytoplasm in our plants, the system is really smart, and it automatically will start looking. Again, we're doing this on a low resolution image. It then will use that drawing criteria that we gave it to take a high resolution image only in the areas where we drew. And then it'll do it over and over and over, literally thousands of slices, where I can only image at high resolution, which is the slow part of a scan in the areas of interest. So we've seen savings well over 50%. Depending on how much biology of interest is in that particular field of view. When you're finished, you have a low resolution image, which is very quick to take, but still has enough, let's say our pixels are 50 nanometers, enough information of the rest of the context of the cell. So we maintain the low resolution context, and we can embed the high resolution image directly on it. So number one is we maintain context. Number two, if I'm doing whole cell imaging at nanoscale, maybe 5 nanometer isotropic voxels, those are thousands of slices. I think a lot of scientists would typically not want to go forward with that because of the time it takes. Well, if I cut the time in half to do that, something that would take several days would take much, much, much less time, half the time, which means I can either do twice as many cells, or I can reduce cost of running a particular project and be more efficient with it. So I think probably the biggest thing for volume EM has been, statistics, low numbers of N. I think this will go a long ways towards it. And it'll also make it more affordable for investigators to come in and start a volume EM project because you can get twice as many samples done, or you can do lots of little different regions and then focus just on what you care about in the image and still maintain some context at a little bit lower resolution.
Really, it was a pleasure working together with Kirk, developing this adaptive scanning approach. But did he share any insight? What was the change for his scientific question? Is it just the technology that makes it easier and faster, or was any clear benefit that he articulated about science?
I mean, we talked a lot about the different projects that he has on the go, and one of them in particular, actually, that he spoke about in relation to adaptive scanning was associated with plasma-desmata.
In our work, there's two areas of high interest, and one of the things that's driving more efficient scanning is that every plant cell, almost every plant cell, the vast majority of plant cells are connected to adjacent cells by small pores called plasma-desmata. And these pores are around 40 to 50 nanometers in diameter. And so you need an electron microscope to see it. And it turns out that when you look using a transmission electron microscope in an ultra-thin section, you see a handful of them at a time in a field of view. But after taking 1,000 slices, it turns out that we're regularly seeing many hundreds to well over 1,000 plasmidesmata per cell. Turns out that every cell inside of a plant is connected to adjacent cells through these channels. And so you can, from the root to the tip at these plasmidesmata, all these cells are connected. So they're signaling each other. But also for the work that I do, pathogens use them as a way to get into the next cell and cause disease. So we need to use electron microscopes to be able to see these things, challenge them with pathogens, and go in and actually see what changes are being made. And it can only be done now, as we're looking at it, we think that 2D is probably not the best way to make gains in this field. We think that 3D is providing critical information.
During the collaboration, we have a lot of discussion about volume imaging, but not only room temperature, also cryo. Did you talk with Kirk about his aspiration in cryo imaging?
Yeah, again, we had a really good conversation about cryo, and he shared some really interesting insights, actually, about the future as he sees it for cryo imaging.
It turns out that, we have probably right now, since we've gotten our system, we probably have half a dozen grants that are in to do volume electron microscopy in general. But the ones that are most exciting to the review panels is our work where we're doing cryo, because it's still very young field, and cryo-ET is relatively established. When I say cryo-ET, I mean cryo-electron tomography. But in situ cryo-electron tomography requires us to go in and extract materials from the cell in a very precise location. A lot of our structures that we're looking at have fluorescent proteins or some fluorescent component to it. And when you're doing cryo mode, you have to be able to find your target. And we often use the integrated fluorescence light microscope that comes with the Hydra to locate those things and get to them very quickly and efficiently.
That's a very useful add-on. You can be more efficient and you can avoid annoying artifacts like contamination when you're transferring a sample from an external light microscope to your FIB-SEM tool. What I particularly find important is that you can really navigate and target your region of interest in such a way that you can have step-by-step confirmation of your region of interest while you're preparing the lamella. This means that by the end, when the lamella is already finished, you can still confirm that your signal or your feature of interest is still within the lamella. That makes the efficiency when you're going to the TEM really, really much higher. So I find this very, very useful, as Kirk noted, navigation and identification of your region very fast and you focused.
Yeah, absolutely. Loads of potential, both for lamella prep and for volume acquisition, being able to target specifically what you want to see using fluorescence. Very cool. So in terms of the cryo functionalities, is there any additional things that you want to talk about? We've spoken about the IFLM, we've spoken about the capability to capture volumes and lamellae. Anything else you'd like to touch on?
So what are the challenges when we talk about cryo acquisition? It comes with its own issues, and they're very simple. They're connected with the charges, they're connected with the sample morphology, they're connected with the fact that you are really using low current, so low contrast. All of this is giving a hard time to an automated systems, which means that your auto functions like autofocus, auto contrast can really fail. What we do now is we just, again, like you have seen on the video, annotate a specific feature. This feature is automatically segmented by the algorithm with each subsequent slide. And what this feature does, it is just the ruler for where your auto functions will be placed. So they're not lost, regardless how the morphology of the cell is changing or of the sample that you're looking at, or if you have some kind of challenges with your contrast. It will ensure that you will have these dynamic auto functions and they will be always on the right place.
Maybe if I go back a little bit to Kirk now, he was talking about cryo-imaging, he was talking about room temperature imaging, but he wears a lot of different hats. He runs his own research group, and he's also head of the Advanced Imaging Centre there. And I was kind of interested from a usability perspective, with all this functionality that's available, his perspectives on the Hydra Bio in a core facility environment.
The system is just highly versatile. And, when you're working with either a large or smaller core facility, for example, being able to take a single platform and have all of these tools that I can use to solve their scientific questions is very powerful. So I do appreciate this particular platform for its versatility and the types of experiments that we can run. It means you got to be, know a fair bit about all these different things, but the resources and the capabilities are there. And I expect we're going to have a lot of exciting research and papers coming out from this capabilities.
It's very nice to hear the success that Kirk's already having with his system. And this adaptability of the Plasma-FIB is something that I also heard from Jirko Novacek, who runs the core imaging facility at SciTech.
Our facility serves an open access hub for high-end structural and cellular biology research using electron microscopy. The facility works for different types of users, including the internal users here at SciTech or the academics from all over the globe. And we also have a collaboration and to support the industry users.
It's a really impressive setup at SciTech, I have to say. Another great conversation. And I asked about the range of specimens that they see coming to the facility.
Within our facility, we encounter different types of samples, starting from the purified protein and protein nucleic acid complexes that are then studied by single particle analysis, over to more complex samples such as whole cells and tissues.
I asked specifically about the Plasma-FIB and the kind of experiments and research questions that can be addressed with the system.
I mean, from the perspective of the facility manager, I see a Plasma-FIB-SEM instrument and a critical enabler for many of our users. It is actually not only the FIB part of the instrument, but what I really see as an important component is the high-quality cryo-SEM imaging that we receive when using these tools, which in a way is also very, very important for the lamella preparation workflow for cryo-ET, where it can partly or completely substitute the need for correlative fluorescence imaging for within the targeting workflow. These tools in our lab are primarily used for volume EM work. This is both at room temperature, so for in-resin samples and reconstruction of the whole cells and parts of the tissues, or then under the cryo conditions, again for cryo volume imaging of cells and tissues. And there I see the combination of the Plasma-FIB with the high-resolution immersion SEM imaging very important as particularly in cryo volume imaging, we still need to detect any electron that comes to increase the sensitivity. And in our own hands, this was, for instance, shown when using oxygen for the milling and increase the contrast in the eventual cryo-SEM data.
I know the magic of oxygen for room temperature, but I believe for cryo we're still exploring which is the optimal ion beam for which type of samples. But great note from Irka.
So cool. There's so many different things still to explore with the technology.
What I find particularly exciting about cryo volume imaging is that it is currently a developing technique, and we still do not see where this leads us. We currently know that we can obtain sub 20 nanometer resolution information from the data, but we still not it's still not obvious whether this is the limit or we can go further. And I personally believe that with the advancement at both the technology and the workflow side, we will eventually show that we can reach high resolution to see more biology in the context of the larger volume of the biological sample.
What a nice way to finish. Seeing more biology in the larger context of the biological sample. It's a very fitting way to summarise the possibilities of Plasma-FIB and the Hydra bio in general. So we're now going to move on to the live Q&A part of the webinar. I'd like at this point to say a huge thank you to all the researchers who were kind enough to share their thoughts and their experiences about the technology. If you have any questions on the technology itself, on the research of those involved today, or you would like to know more, please do reach out to us. So without further ado, let's move to the live Q&A. Hi everyone, welcome to the live Q&A. Thanks again for joining for this session. Please continue to submit your questions. We have got some time. First and foremost, the results with the oxygen milling looked amazing. Is this always your go-to protocol for cryo-EM imaging? Jyrki, that's your work. I'll come to you.
So yes, currently we primarily use or solely use oxygen for the imaging. However, for the preparation of the sample, we use xenon because it's faster.
Fantastic, thank you. And then maybe to Maud, on a similar vein, how can you make sure you match a sample with the best ion combination?
Yeah, good one. In addition to what I've been just said about the contrast and the imaging, I think what is important for us also is the curtaining rate. So, and how flat is going to be your surface. It's sample dependent. There's plenty of things. It's not only the ion source, it's how you prepare samples. You know, there are tricks to prepare very flat surface by preparing first a flat surface and then use this first surface to put your protective layer and then mill this one as a front. But in addition of that, you may want to check, and that's something that we've published in eLife, your proportion of having some curtain form depending on a combination of current and voltages as well as ion source. So a bit of a mixture of everything to explore here.
Excellent, thank you. We have a question on transitioning between cryo and room temperature. Jyrki, you mentioned that in your facility you do both. The question is, how easy is it to transition the system from cryo to room temperature, and is there significant downtime to make this transition?
No, not really. At least not that we would see that. Typically, I mean, we do have cases where we continue with the room temperature the day after the cryo session. So we typically leave the overnight for the system to recycle. But other than that, we have not seen, if the question was aiming the other way around, we have not seen an increased contamination rates on the cryo specimen following the room temperature session, if that's where the question was going.
Fantastic, thanks, Jyrki. Excellent. Tatiana, you spoke about lamella prep in your section. Does high current Plasma-FIB cause issues with lamella prep?
So to be perfectly honest, we use the high currents to preparing our area of interest where we want to place the lamella. For the lamella prep itself, for the rough milling and the polishing later on, we use similar currents that we would use for gallium as well. So there we do not use these high currents. To make a long story short, and please look out for the paper that is going to be published in hopefully soon. We found out that with these extremely high currents that are available, so for example, for xenon 2.5 microamps that are available, you can use them for trench milling on your sample. You just need to be aware that there is going to be some sort of damage. A certain distance of the or a certain volume of the sample is going to be damaged. If you take that into account and remove this damaged area with a lower current that will not introduce any additional damage, then you can totally fine use even these ridiculous currents up to 2.5 microamps on your sample. And then you get beautiful lamella in your area of interest, and you can use these high currents for trench milling to access your area of interest.
Fantastic. Thanks, Tatiana. A question around sample preparation for adaptive scanning. Kirk, how does the sample preparation impact on the use of adaptive scanning? I guess we're at room temperature now.
So if you're doing traditional volume EM and resin and you're using your metallization protocols, I think the main criteria is to make sure that you have your object is clear and has a defined boundary so that it can find the edges. So normally that's okay when you're looking at a cell versus a non-cell. If you have mitochondria, for example, with high contrast inside of the cell, those should be relatively easy. So it's about having, clear boundaries. If you have low contrast, it'll be a little harder. My samples, work perfectly fine under those conditions, and we've tried a couple different ones for the paper as well that you can look at. So it doesn't have to be perfect, but it does a really nice job. You just need to make sure the contrast is for your object is there.
Fantastic. Thanks, Kirk. Sveta, one for you. Will Thermo Fisher introduce interleaved SEM scanning patterns to reduce charging artifacts? And this refers to Maud's work.
Yes, absolutely. So we are looking and exploring the options, and we definitely keen into finding a way to integrate it into the platform. So work in progress.
Fantastic, excellent. And then one up for grabs, actually, a BSL2 question. Recently on an EN forum, someone was asking about how one can use these techniques for BSL2 or higher samples. Do these scopes have to be in a BSL2 lab to look at such specimens? Who'd like to take that one? Volunteers.
I'm afraid it will depend somewhat of the legislation of the country that you're in. So it's a bit difficult for us to answer generally and broadly to that. So I would refer to your country requirement, but there is also a discussion that you need to have with your thermo engineer team and your field service engineers to agree on the level of service that you're going to have afterwards, irrespective of the country where the microscope is.
Okay. Fantastic. Thanks, Maud. We have a question on materials-based research. Is there an issue with different types of materials-based research being used in the Hydra Bio? Sveta, maybe if I direct that one to you?
I'm not sure what material-based research, if it's material science, I don't think so, but we have to be fair and we have to state that Hydra Bio was really optimized for the life science research, and the challenges, for example, with cryo or focusing on things which are typical life science. So yeah, maybe the complete range of material science research might be a challenge when you want to use it with Hydra Bio, but this will very much depend on case by case. So, but in general, no.
Fantastic, thank you. Another question on lamella prep. So Tatiana, how do your lamella prep workflows differ now that you have Plasma-FIB?
They are essentially the same. So there is no difference in routine. We use either, depends, so we have some users that prefer milling their lamella by hand, but we also use all-the-time cryo on the Hydra Bio. Our own personal preferred way is doing rough milling on the lamella with xenon, and then doing the polishing with argon, but you can also do the polishing with xenon. This is completely up to you. You need to get a teeny tiny bit of a feel for how the beam behaves, because the beam diameter is going to be bigger on the Plasma-FIB than what you're used to on a gallium FIB. But other than that, the approach is super straightforward, and if you can mill on one type of instrument, you can, not just milling, but also do lamella polishing on one instrument, you can do it on the other instrument as well. So there's no change in procedure that we have.
Fantastic. And maybe sticking with that, we have a related question for lamella prep. How does the consistency of lamella thickness compare between the two instruments, the Hydra and the Aquilos, in your experience?
So in our experience and in our hands on the Plasma-FIB, the lamella have a tendency to be a teeny tiny bit thicker So our thickness is in the range of 150 to 200 nanometers. So maybe a bit thicker than you would expect on an Achelos, but super consistent, no problems, very straightforward.
Fantastic, thank you. On to a cryo volume question. Mort, I'll come to you with this. How do you prepare samples for optimal cryoimaging?
Oh, it depends on the thickness of your tissue to start with, or what you want to look at. If we go for the rule of thumb is that anything that is beyond 10 microns should go, thickness I'm talking about, should go for high pressure freezing, and anything under may be able to go for plunge freezing. All of them may or may not required some addition of cryoprotectant. So you will find from the Franklin, but many other places, detailed protocols to explain you how to do and what to add to help you to have the best vitrification possible. I think that answers the question, I hope.
Yeah, absolutely. Very varied, I think, depending on what you want to see. Fantastic. And then Kurt, sticking on cryo, why is cryo volume imaging so important? You mentioned during your part that was a really interesting area.
So I'm going to be the first to say there's a time and a place for resin or cryo based protocols. And some questions, the advantage of cryo would be for very dynamic structures, but also you cannot underemphasize the importance of the fact that your sample is vitrified and hydrated in its native state. And of course, then we get all the benefits of structural biology. So we can't rule that those kinds of measurements with plastic. There could be some changes. You're using metal to get the signal. This is all contrast that comes from the sample. The only pretreatment is what you do to get it frozen. And you can get remarkable contrast. And so my comment is there's a lot of biology where you will be able to reach that you can't maybe with more traditional volume resin based.
Fantastic. The future is definitely bright. Jirka, you're a structural biologist. Why did you start to explore options in room temperature? It's interesting that you have such a combination in the facility now.
True. It comes from the users. So this originally started serving the structural biology community, but as the time goes, that community starts to try answer questions that are more towards the cellular biology, so more contextual. And this all of this Lamella workflow, this is in situ structural biology. And then we're one step towards the ultrastructure. And despite cryo volume is actually nice and probably future, for many cases it might be just simpler and more straightforward. And there might be more workflows for the sample prep for the in-resin work. So it was a logical step to adapt that as well.
Fantastic, thank you. Another question on the AI. Sveta, I'll come to you. How can you be sure the AI recognizes the specific structures of interest?
It's because of the genius team that we have. Now the algorithm is designed in such a way that it can tolerate some kind of a over-segmentation. So we know that it's very important that you're not losing the region of interest. So you don't want to have any miss of information. It's, as Kirk already mentioned, it could be challenging when you have a very complex, dense structure and the AI needs to find your structure among the rest of the contextual volume. But in general, it's a very robust, and you can even adopt on the fly. So usually you will have a very long acquisitions and you might just stop after 2000 slices and adjust your mask and tailor it further. or even add a new feature if a new feature of interest was occurring in the volume. So it's very adaptable, and we definitely take care that you have only over segmentation but not losing area of interest.
Fantastic, thank you. So we're rapidly reaching the end of the session. I'd like to say a huge thank you to the panel for being here today and for all of the scientists who've been involved with this endeavour. It's great to talk about this technology, all the exciting science that's going on. If you have any further questions, please reach out to us, and we'd be delighted to put you in touch with the right people. So thank you so much for joining, and hope to see you at the next event.